recording maybe because I did stop recording. Okay, so we've got Lisa, Lizzle and Doug on the call, Michael, Brett and a couple of other people who I don't recognise. But we will...
Hey, it's me, Mohammed.
Mohammed, how are you going?
We've got Diane.
And Diane in Prosy.
Yep.
Perfect, no problems. I'm just going to attempt to show you my screen. How are we going there?
All good? I can see it.
Perfect. Okay, well, thanks for coming along. I don't know whether you can see me too, jostling around, or you can just see my screen.
We can see you too.
So how do I mute that? Because I really want you to just be focused on the screen, not me. I have... what do you need to do? This is my Justine here, if you can see Jane.
What do you want me to share?
Just to delete me being video. So we had that on mute before. I'm just using Zoom rather than Skype because it seems to have better connectivity. There's video.
Okay.
And resume share. So you should be able to hear me.
There we go. And close it up.
Okay, so how's that? Can you just see the screen now? Hello?
I seem to not be seeing anything at the moment.
Oh, you're not seeing anything?
We can just see the screen.
You can just see the screen?
Yep.
Ah, there we go.
Okay, so we'll just make that bigger. Can you just see the screen now?
Yes.
Yes.
Beautiful. Well, I will start. Thanks for your patience. So my understanding is we've got an hour to do this, so I'll go through. So the purpose of doing this is really around some of the practitioners feeling a bit uncertain with their understanding either of chiropractic and the science behind chiropractic, or their understanding of current research that's really supporting a lot of what we do. And so I thought I'd share some of that with you because I know it's very frustrating and can be isolating when you're in a practice by yourself and you're seeing a lot of patients and you see things in the media that perhaps aren't supporting what you're doing, or they're supporting other modalities that are supposed to be instead of being someone seeing a chiro. So I'm going to go through essentially what it is that we do. So the housekeeping, obviously get comfy, make sure you've got your mics on mute unless you're going to be talking. That's just so that we've got good clarity and no one's listening to you eat your lunch or do the dishes while this is on. And we can ask questions throughout it or at the end, whatever you want to do. You can, if you type them to me, I probably won't see them. So just unmute and ask me a question. Okay, so the agenda, we're going to go through some recent chiro research. Some of the chiros who've been in TLC for several years may have seen a little bit of it, but I'm presenting some of the stuff I've given you previously alongside new information as well, in relation to the adjustment, what we know about it, and the neuroscience research behind that, the research behind the dose response and how often we should be adjusting our patients, and any supporting research for adjusting children, and using the SD protocol, which I know a lot of you understand the basics of the SD protocol. So there's some really great research coming out to support that work as well. So we'll just head off now. So obviously we want all our patients to look like this little guy, very happy and loving life, getting an adjustment. So one of the things that's been very recently published, and this was a study by Chris Chen in Nature, which is obviously, it's Nature Reviews Molecular Cell Biology, I beg your pardon, not just Nature. This was recently published last year, and it's talking about changes in the cells themselves, because cells have fibrous connective, not adhesions, but more like very fine fibres that are within the cell themselves and outside the cell. So I'm going to show you a very short video that explains what this actually means. So hopefully, fingers crossed, this all works. Can you still see my screen? So it goes for 6 minutes, but it's about how the pressure of adjusting someone can affect the individual cells. And it's the speed
My
of the
name's Don
adjustment.
Ingber. I'm a senior author with Akiko Momoto and Tada Momoto of an article that is a review on the role of mechanical forces in development and disease. And it's a little unusual for a biological review article because most of the work in the last 30 years is focused on genes and chemicals and hormones. But my lab has been working for 35 years and more on the key role that mechanical forces play. And what we found, really to cut to the nutshell, is that mechanical forces are as important for control of cell and tissue and organ development as chemicals and genes. And so we feel like this review, which we tried to make as expansive as possible, provides a new perspective on how cells are regulated by physical cues from the environment. These are forces like shear stresses in blood vessels, blood pressure, but also when you move your arms or you have stretch of your skin and your muscles, or gravity on bone and cartilage, but also down at the microscopic level, the forces that cells generate inside their cytoskeleton, the molecular framework of the cell, those are equally important in regulating cell and tissue behaviors. So all cells have this cytoskeletal framework, which contains actomyosin filaments, which are the same actomyosin filaments that we all have in our muscles when we contract our muscles. And when cells contract, they pull on their adhesions to other cells and to the extracellular matrix on which they adhere. That's the in vivo anchoring scaffold. I like to think of it as an egg carton on which the cells sit like eggs. As the matrix orients the cells and provides basically a scaffold for their orderly renewal and repair. And so in this article, we actually start at the beginning of development, from fertilization of the egg all the way through the divisions of the cells in the early embryo to the formation of the first tissues and organs, to the morphogenesis, the generation of form of the particular tissues and organs.
Can you just mute your mic? Sorry, I've got someone with a fair bit of work they're doing near their mic. Can everyone just make sure your mic is muted?
Then how tissue architecture is maintained throughout adult life, and how mechanical forces actually govern some of the major control points at all of those stages, and then how when the mechanical sensing and transduction systems, we call it mechanotransduction, how mechanical signals are converted to a chemical response, how that system sometimes can become deregulated, you actually can get disease. Examples of mechanical forces in development. We've done work in our lab recently where we found, for example, that the formation of a whole organ, in this case a tooth, you don't think of it as an organ, but a tooth actually has different tissues within it that interface, lining cells called epithelial cells, and then mesenchymal connective tissue cells, fibroblasts-like cells on the opposite side, that start out as a simple bud off the lining epithelium in the mouth in the embryo, and then this bud buds out, so there's a little, the bud sticking out, and then it buds in on itself, so it forms two little fingers downward, which are the roots, and that's all of morphogenesis in that tooth. Blood vessels come in, nerves come in, the epithelium differentiates into the hard enamel and dentin of the outer covering of the tooth. And for 20 years, people have been working out all of the genes and the morphogens, the chemicals and factors that regulate the steps that go from that initial bud to a tooth. But no one really understands how it works. And what we found, and this is just one example, is that the cells that are on the upper layer, the epithelium, the lining cells, put out two different factors. One, induce cells to migrate towards the the matrix on which they sit. So the cells beneath are induced to migrate upwards towards the epithelium. And that's at a very gentle gradient. So cells all the way in the distance are starting to march towards it. And at the same time, the same cells produce out a factor that's a repulsive factor that basically tells the cells that are coming close to turn away. And this causes the cells to compact, to crowd together. It's called mesenchymal condensation. And this is known for probably 50, 100 years, that there's a mesenchymal condensation as a first step before most organs form. And in fact, the shape and size of that condensation can affect organ size and shape. And what we found is that that physical compaction, the compression of cells tightly packed, is enough to trigger the transcription factors that induce all of the differentiation steps that lead to tooth. And you could actually take this undifferentiated connective tissue cells and physically squish them between two pieces of rubber, and they can then be induced to go all the way to a tooth when combined with the epithelium. So that's a case where all the genes and chemicals that people mapped out are indeed correct, but the genes encode for these motility factors, which then manifest their activity, their form-inducing or organ generation-inducing activity, through inducing a physical change in the microenvironment. And then once the cells experience that, you don't need the factors anymore. So I think there are many other examples in the review, but I think the important message is that we need to take into account physical forces, physical cues in the environment. We need to develop model systems to be able to perturb those physical cues, control them as a separate variable, to really begin to understand developmental control.
So that was certainly an interesting video about that. Sorry, I'm just going to get you back to where we were. Have I got everybody back online? Just someone say yes?
Yes.
Excellent. Thanks, Lisa. So I'm hoping that you saw the video, because it's very exciting that there's research that demonstrates, obviously, that the adjustment, the power of an adjustment and the pressure of an adjustment can actually change cell structure, can change genetic expression. And this study here, of course, was published some time ago in 2007, but it's looking at the effect of muscle spindle afferentation, so an adjustment, on changes to the function of the nucleus tractus solitarius. And if you recall your neurology, the nucleus tractus solitarius is actually the birthplace of the vagus nerve. And the vagus nerve, of course, is the main nervous system supply for our organ systems. So, and obviously the sympathetic drive comes from higher up in the brainstem, from the mesencephalic reticular formation is the primary sympathetic neurons, and the secondary ones are in the thoracic spine. So when you're adjusting the cervical spine, you have a direct influence on the nucleus tractus solitarius, which is something that we often notice in practice, where you can actually perform an adjustment and someone over time will have changes in the way that their organ systems start working. The work from Pelleccia in 2015, his paper published in the journal Musculoskeletal, is neuroplasticity in the central nervous system the missing link to our understanding of chronic musculoskeletal disorders. So what he found is that when there was changes in the way somebody functioned, if there was a chronic pain, or not pain state, if there's a chronic malfunction with the way that a person moved, then what happened was that there was an actual change in the way the brain then continued to function. So it's a matter of, if you don't use it, you'll lose it. So if you're not moving joints, if you're not moving muscles, if you're not moving the body as it should be, if there's an asymmetry anywhere functionally, then your brain will start to transform the way that it functions. And we know that in a lot of the work that Rob Malilo does, we actually see that in the research papers that he has with the hemisphericity model, and Ted Carrick has with his, and Brandon Brock. So there's a lot of work those guys have done into seeing, well, if you injure your right arm and fall over from the trampoline as a 5-year-old and you have your arm in a sling for 6 weeks, that left side of your brain isn't getting that stimulation that it should be. So is that setting you up for a left-sided hemisphericity? Should we be making sure that after you've had that type of injury, we really focus on rehabilitating your brain after you've had a repair to your arm? And the same obviously is true on a much smaller state in the actual joints themselves along the spinal column. And so I think we've got a very exciting job. Alterations in cortical and cerebellomotor processing. So in subclinical neck pain patients, these are people that have no pain, but they have malfunction in the way the joints are moving. So they had 10 volunteers. This was a study done by Delicto in 2013. I think it was published in GMT. I can get all the information for you and send it to you. They had 10 volunteers and they tested them before and after an adjustment, and found that when they measured cerebellar function, the people who had subclinical neck pain, so in other words, they didn't have any pain, but when they were palpated, they had poor movement. So they were adjusted and they found that they improved in their cerebellar function in comparison to healthy controls who didn't need adjusting. So this is where you get the question, when should I be adjusting patients? Well, when they've got a subluxation, when they've got an aberration to the way their joints are moving, they should be getting adjusted. Now, later on, I'll show you a dose response study that was also done about how frequently, because obviously it would be possible, or is it potentially, too much? So there are studies done on that as well. So we'll go through that too. Again, this is another subclinical neck pain study demonstrating mental rotation tasks. So again, this is 26 healthy volunteers. This study was again done in 2015, published in JMPT. Heidi Holvik also worked on this study. So it's showing that subclinical neck pain impairs the ability to mentally rotate an object, and that works with your parietal lobe, because obviously your parietal lobe is where you store somatosensory information and images, particularly in the left parietal lobe. So the study that I've been, that's on hold, that I've been doing with the New Zealand College with the brain scientists there, so Heidi and Kelly and some other neuroscientists, is looking at this same type of study with children. We haven't looked at the results yet, but we've done the actual study, and we're predicting that it will be the same result, that will show that children who have problems with the way their spinal joints are moving don't mentally rotate very well, and once they've been adjusted, that improves. This is another study by Kelly Holt is involved in this. Heidi, Dina Lalick was the main researcher. But essentially, again, we're looking at if spinal function affects anything else, and what we're showing is it affects sensorimotor integration in the prefrontal cortex. So remember, the prefrontal cortex is where you have your ability for your cognition, your ability to drive. A lot of that frontal lobe is in that prefrontal cortex. So your understanding of information, your executive function, your working memory, all of those areas of your brain can be underfunctioning. And what they found in this study is once a single session of, they call it spinal manipulation all the way through, we'll call it an adjustment, of dysfunctional segments in subclinical pain patients, changed somatosensory function, which is pretty phenomenal. If we don't want to go along the neurological pathway that is certainly what the breadth of chiropractic is about, which is what we saw with that first video on Dr. Don Ingbar, the study which showed the forces and changes to the cell. If we just want to market ourselves as back pain specialists, which I would strongly encourage you not to do, but should you want more information about how well we do there, this is a guideline published just recently by the Canadian Chiropractic Association, and they're showing from a oh gosh, I've forgotten the name of it, a study that looks at all the possible papers that are out there, that there's spinal manipulative therapy is the key form of treatment for either acute, chronic, or chronic pain, back pain with radicular symptoms, and that this is what they're sending out to all of the medical practitioners around Canada to ensure that the most highly regarded form of therapy is applied. And this is what has been found in this particular study. The cost effectiveness of spinal manipulative therapy. So again, this is one I showed you guys a couple of years ago. People who are doing home exercises plus getting adjusted have costs that are up to 47% lower than people who are just doing standard rehabilitation and their own exercises at home. So you're 47% more effective than people just doing anything that they might do at the gym, or if they've just been shown exercises by their GP. Upper cervical and upper thoracic manipulation versus mobilisation. So mobilisation has been statistically more strongly used by physiotherapists. So this is a study in 2016 which showed that of the 110 patients, where they compared high velocity, low amplitude thrust, which is what chiropractors generally use, compared to mobilising and just giving exercises. So we found that 6 to 8 sessions of a high velocity thrust was more effective than mobilisation. And this was in relation to patients with chronic headaches as well, who had their cervical spine adjusted, that maintained that gain over a 3-month period. So it was much longer lasting than obviously the common Panadol that most people reach for. Variations in patterns of utilisation and charges. Again, this is another study published in 2016 in GMT. So what they found here was they were looking at complicated or uncomplicated neck pain patients. So where patients had radiculopathy or headaches associated with it. So what they found essentially at the end of this very large study was that patients who saw the chiro were between 54 and 84% cheaper in costs than those who went the route of seeing their medical practitioner or their physical therapist. This is a US study, obviously. And so in America, you don't have free medical care. So all of the costs, including the visit to the GP, as well as the medication, the time off work, all of that was factored in to the chiro, the GP or the physio, and the chiro care was at least half, if not more than 80% less expensive for patients suffering neck pain than any of those modalities. Some of you also use the SD protocol. So the SD protocol obviously has, I'm hoping my meme is working. I can see it on my screen is working. My GIF, I think they're called. So, it was, we all have patients that aren't necessarily coping very well, and some of them have what we now know as the SD symptoms. So what we find is that patients will have a increased stress response. They'll often be in a fight-flight response, so their pupils will be dilated, the muscles in their neck will be tightened, the muscles in their pectoral muscles will be rounded, their shoulders will be much tighter than usual, their thoracic cord will be wound up, the sympathetic second-order neurons. They'll have firing into their buttocks, and their low back will change its positioning, so they'll often have a hyperlordosis as well as that, because when you're in a state of fight and flight, your parasympathetic system gets shut down, so your gut tends to become leaky. You can have an increase in your heart rate, you can have an increase in your respiration rate, in your sweating response, and you'll have a decrease in your sex drive and a decrease in your reproductive system. You'll also have a change in the way your cortisol level fires and the way your thyroid functions. Excuse me just for 1 second. Can you just go to someone else and talk to me? Sorry, I've just got someone to move. So the other thing is that you'll have heightened cortisol levels, which will affect the way the thyroid functions, the way the amygdala functions. So you'll have reduction in thyroid function, weight gain, thinning hair, brittle nails, dry skin. So this research is in relation to that. And obviously as chiropractors, we can do an awful lot of work to improve the function of patients who have the myriad of symptoms that line up to be SD. And that's what Wayne's book is all about. So there's a 7-step process that we can go through to assist patients to have a better functioning body, and obviously the adjustments that we do, the standing ribs and the resetting the first ribs is one of the biggest first steps of that. And as chiropractors, we certainly are one of the only professions who do that to reduce cortisol levels in the brain. And the other things that we're doing obviously is lifestyle changes that we encourage patients to do. So one of the things that we find in people with wound up sympathetic nervous system is that they're more likely to have a heightened heart rate. So what this study finds, because a lot of people with SD actually have autoimmune diseases, and rheumatoid arthritis is considered an autoimmune disease. And this paper goes a little way to explaining how that they may be linked. So it's demonstrating obviously that people who have a tendency to develop rheumatoid arthritis are those people who have a genetic prevalence for it, but also that their heart rate is actually at a baseline level significantly higher at resting state than the average person. So those people with a resting heart rate above 68 are more likely to end up in the rheumatoid arthritis group than those who have a normal resting heart rate. So you want your resting heart rate to be closer to 60 and certainly not above 70. Oops. This study here, and I'll have to find you the references because I put this one at the last minute. This study here shows the changes in gut flora. So one of the things we talk about with SD protocol is that as your parasympathetic nervous system is functioning less well because your sympathetics are in overdrive. So in contrast, it seems that the parasympathetics shut down. You tend to have greater permeability to the gut itself, and so your gut doesn't tend to function very well. So what we've often found as well is that we need to replenish the gut flora in people that have SD. And whether it's the gut flora that is a problem initially that sets them off, because there's actually some really interesting research that's coming out now about children in the autism spectrum and their gut flora being a problem in the beginning that sets off their brain-based changes. So this study as well, which doesn't talk about autism spectrum or ADD, but it talks about the HBA axis activity and how that changes in people who have an unhealthy gut. They have an increased anxiety and stress response. So changing their gut flora will help to reduce the stress response, which is why it's important when someone does have sympathetic dominance that you don't just adjust them, that you make sure that you're looking at ways to calm down their sympathetic drive by getting their gut flora to calm down. There's another study, and again, I apologise that I don't actually have this for you, but it is a study showing the changes to how the Edgar-Westphal nucleus changes how the amygdala responds. So you recall when someone has a hemisphericity, which is based on the higher cortex not functioning very well and being able to dampen down those mesencephalic nuclei. So the Edgar-Westphal nucleus is one of them, and in SD protocol we see a lot of people with dilated pupils because their brain has been in a stress response for a long time. So what we're showing and what we're finding, sorry I've misspelled normal there in my enthusiasm, is that we've got a change in the way that we can actually respond to stress because we've got that Edgar-Westphal nucleus being chronically stimulated. So what this research was showing is that the Edgar-Westphal nucleus may be priming the amygdala and not the other way around. So we want to do whatever we can to dampen down an overzealous Edgar-Westphal nucleus. And we know that by stimulating the higher cortex, which an adjustment does, if you recall the neurology behind that, when you perform an adjustment, especially at a speed of greater than 120 newtons, milliseconds, I beg your pardon, you're going to stimulate and afferentate higher up the spinal cord into the brain. If you do slow stretching mobilization, you only get a local effect. So the effect of an adjustment into the spinal cord affects the spinocerebellar pathways. Those spinocerebellar pathways go into the cerebellum, and the fastigial nucleus in the cerebellum is the main nucleus that an adjustment affects, and it goes across into the mesencephalic part of the brain, or into the brain stem, and we have the vestibular nuclei. And if you remember your neurology, the vestibular nuclei gives birth to the vestibulospinal tract as one of its jobs. So the vestibulospinal tract holds our neck in good tone, so we have good posture for our head and neck control. We also have the reticular formation that gets messages from this fastigial nucleus in the cerebellum from our adjustment. So the reticular formation is the birthplace again of that reticulospinal tract. So the reticulospinal tract gives us tone to our trunk. So we're going to find that an adjustment fires that pathway to give us central tone. So when you've got a baby with low tone, an adjustment is firing up all of those nuclei to encourage the restoration and development of good tone, which is really important for a baby's development. What else was I going to say? The other nuclei, of course, that's there is the nucleus tractus solitarius, and the fastigial nucleus fires from the cerebellum into that after you've done an adjustment. And again, that's what we saw with the earlier paper, where we're looking at the effect of the nucleus tractus solitarius being fired upon from the effect of your thrust and your adjustment through the spine. This is another study published, where was this published? In 2013 in a clinical nutrition metabolic care journal. So this paper talks about a hormone that's in the gut called ghrelin. And ghrelin from this study has quite a strong role in sympathetic nervous system activity. So again, it's about restoring the gut function, because there's from this paper a very strong connection with how healthy your gut is and the microbial bed that is what makes up your gut, and to how well your ghrelin can function. And the ghrelin actually, when it's out of balance, can affect how your sympathetic nervous system works. It affects how the heart works, the bones, it affects everything, but it certainly has a direct connection with the sympathetic nervous system. So how often should we be adjusting patients? The conclusion of this paper, which was published in 2013, was a study looking at a variety of different dose responses, and they looked at 6, 12, 18 and beyond adjustments in a short period of time. So I think it was over a 6-week period of time. Sorry, it was at 12-week and 24-week endpoints. So what they were talking about was that the dose response was that you needed to have delivered at least 12 visits within that 12-week period of time to get the most favourable results. Those that were above 18 had no better response. So there was between 12 and 18 visits in that first 12 weeks has the largest and most long-lasting effect on restoring function, on reducing lifestyle factor surveys that were filled in, on reducing the visual pain analogue scale. And that was more long-lasting when it was delivered in a short space of time. So patients who had less adjustments in that same time frame, like less than 6 adjustments, had far less effect and far less benefit to all of their normal lifestyle factors. So I don't think it's an issue in the majority of practitioners' practices, but there's certainly it's good to go back and review to see if your patients are actually following through, to see whether you're getting the best benefit. So here's some papers on children. I actually had a little starter slide here, but I don't seem to see it. So children in Norway were reviewed. There was chiropractors that were asked to explain a little bit about the patients that they see that were children under 18 years of age. So there was 28,000 people that were reviewed under 18 years of age, and 114,000 adjustments, which goes to show you that they don't really do very many adjustments over there. But what they found was that almost 2/3 of children in the 0 to 4 age group had musculoskeletal complaints, and excessive crying was about 14%. So that's interesting because obviously we often need to support why are we seeing a child. And this is certainly great research that demonstrates that the reason why we're seeing them is because they have a musculoskeletal problem. They have subluxations. If our insurers don't understand what a vertebral subluxation is, we can explain it in terms of function. We can explain it in terms of aberrant movement, in disafferentation, in neuroplasticity terms. Children between 5 and 17 obviously was 80 to 95%, and headaches were the next biggest thing. So again, this shows that on a chart, but it's interesting that so many of the patients obviously had quite a lot of other things, and obviously the newborns and the very young children, it was quite a range of other things as well. This study is back to SD. Sorry that we jumped around a little bit here. So this is a study just recently published again, and I can't see the actual date because I don't have my glasses. Let me just find those. 2016. So what this paper showed was that women under stress had increased levels of cortisol, and the increased levels of cortisol from stress decreases your estradiol production, and the estradiol production is really important for the production of healthy ovum. So the oocytes quality reduced dramatically when there was women were under long-term stress. So again, this is part of the whole SD protocol, the research that helps back that, showing that chronic long-term stress and sympathetic drive, high levels of cortisol, which we can reduce by doing the standing rib adjustment, helps to oxygenate the brain, and that's been clinically shown to reduce cortisol levels immediately. We can help to do that by doing that particular adjustment with our patients and improve their ability to have healthy ovum and hopefully healthy babies. Another study in relation to that was published in 2016, and this was a physiotherapy journal. So they looked at 102 women and found that those with chronic dysmenorrhea actually had changes with the way their pelvis looked on 4D measurements. So they measured how their pelvis was sitting, and if they had distortion or torsion through their pelvis, what they found was that they were more likely to have dysmenorrhea. And there are other studies that have been published, I think from RMIT and also Heidi Horvik, which demonstrate a change in pain levels with women with dysmenorrhea when they've been adjusted. So again, this is the building block with how we have a much broader base of effect than just pain relief in the spine. So children, again, this is a study I've shown you previously, but this is a Joyce Miller study and published in 2016. 85 women were assessed, and the babies were checked to see if they were being breastfed or not. At the start of the study, only 26% of them were being successfully breastfed, and the national average in England is about only 25% of women at 7 weeks are exclusively breastfeeding their babies. So after they adjusted the babies and the midwives were still giving the support to the mums as they did normally, so the only difference was chiros were introduced. They had a whopping 86% of those babies were then being exclusively breastfed compared to those who had no adjustments. So quite an amazing study, and a good solid number of mums and this was a statistically significant change. So really important if you're looking at doing talks to nursing mums. This is a really good study that you can discuss. Efficacy of chiropractic care on infant colic. So this was a study done in 2012. Joyce has done another one on excessive crying, which I couldn't actually, I forgot to put in here. But anyway, there's this one showed that there was a trend for infants to reduce crying time than those in the non-treatment group, and the study demonstrated a change to that in less than 2 to 3 days. So that was a really good, strong trial in relation to colic. Probably the last slide, and I know I've got about 15 minutes before we finish so we can have questions. The last slide that I want to show you is on the accuracy of medical randomised controlled trial results. So there was a study done in 2016 in the journal Trials, and it looked at the different pharmaceutical drug trials that are done, because the practice of medicine itself isn't ever researched, it's the pharmaceuticals that are researched. So how your drugs result in changes. So of 282 clinical drug trials that were submitted, only 72% of them were randomised control trials. Only 39 to 40% of them actually ended up reporting the results. So in other words, what sometimes can happen is if the results don't seem to be going the way that they're thinking, they just don't publish the results. 50% of all the studies reviewed by this particular group of researchers had incomplete data. Their outcomes, their primary outcomes were different to the study design, or they'd changed their sample sizes, which is a big no-no in research, particularly in a randomised controlled trial. And there's a discrepancy in at least half of all published reports. So they either misinterpreted due to adequate or misleading information, and there was a huge risk of bias. And obviously there's quite a lot of the studies that are published could potentially have quite strong bias and a tendency towards results that are not necessarily as clear-cut as they may seem. So that's the end of the slideshow that I wanted to show you. And now if I can go back to Zoom, we can actually start. Have you guys been looking at my screen the whole time? I hope that you have been.
Yeah.
That's okay. So we're basically at a time, I've left 15 minutes so that we can discuss anything else. If there's anything anyone wanted to ask, any questions. Obviously, the more research you look at, if you find a really interesting paper, the more you look at that paper and you go through actually the references, you'll find even more interesting papers that you can build a really good case for. So there's a lot of information that's been published by people other than chiropractors to support what we do. So I'd encourage you to all go out and look for whatever you can there. Anyone got any questions, or are we done and dusted?
These articles in that you've used now to publish, if we want to do talks on them, do we get them from you, or will they be on the Google Drive? And how do we incorporate them into our talks?
We can make them available. I think Lisa has some of them. I can send the rest of them on to Lisa, and she can pop them on the Google Drive, and then you can use them however you may like to in a slide presentation, because obviously some of them are relevant to SD patients, some of them are relevant to mums and bubs, and some just generally when you're doing your spinal care talks.
Okay.
So, you know, there's a lot of really good research. It's actually a really exciting time to be a chiropractor because there is so much neuro research particularly being published all around the world. And it's really important that you keep up to date with it because the old model of chiropractic is really changing and adapting. But I guess that, you know, like I showed you with that forces and cell adhesion paper and the Don Ingbar video, there's an amazing amount of stuff that happens in the cell cytoskeleton, when pressure and forces are applied anywhere on the skin and the body and muscles. And so we're actually changing genetic function through our adjustments, which a lot of, you know, there's a lot of people doing study in epigenetics that support the same thing. So this is looking back at, well, what's the primary factor that affects that? And certainly by applying an adjustment, we're affecting people's genetic makeup every day.
Okay.
Pretty exciting.
Very.
Very. Any other questions? Otherwise, we'll finish up and I'll go have some lunch.
Angela, what would be your best advice to stay updated with the current research? Because there's so much out there, and it can be a bit overwhelming to quite know where to start. What's your best tips to how to stay current on, or which channel, or where to access what's out there and start utilising it?
Well, it is sometimes hard because you can get really good papers when you're at uni because you have access to all of them. But when you get out, it's actually quite difficult to find the full articles. So you can go through ResearchGate. You can go through Google Scholar and put keywords into Google Scholar, and you'll find papers will start to crop up there. And if you go through ResearchGate and open source media, you'll often find some really good articles. When I was doing my PhD, I had the surprise of learning that most of my research supervisors use Google Scholar as their main way of finding articles. And then once they found an article, you look to see what's got the most citations, and the one with the most citations usually is the best written one. And then once you get that, you read through that and you look in the references. And the reference list will give you even more papers that help you kind of explore more that theme. So the papers that I'm going to send you or put on the Google Drive, you'll be able to access that information and kind of explore more about neuroplasticity, explore more about children and chronic pain, or children and getting adjusted, or breastfeeding. So all of those will help. We'll have other papers that will help drive you, and if you'll learn keywords that you can put into Google Scholar, that will help you find sources. But make sure you're trying to get articles that are published in reputable journals, but try and find an open source location of them, because often open source and open access journals can sometimes not be as well vetted, and they're not as well peer reviewed, because you can, believe it or not, write an article and pay for something to be published in some journals. I get asked nearly every second week to put articles in journals, but you've got to pay to put them in there, whereas the best ones are the peer review ones where they're quite thoroughly reviewed, and you're going to have, it's going to be really hard to publish it, and you're definitely not having to pay someone to publish it. Does that help?
Yep, thank you.
Heidi Horvick also has, I think, a lot of research that she reviews, and she puts cute little animations with it, so that makes it easy for people to understand. So I'd look up anything that Heidi's done on your Google Drive, on your Google, and you'll find a lot of papers from her too. Okay.
Just another thing, Angela. the big thing for me is my mind doesn't work in a way that really understands the articles very well anyway. Is there a place that, I don't know, gives it a bit more of a plain English, I guess would be the term?
Yeah, they certainly can be confusing. And often if you just read the abstract, you miss the main part of the methodology that might explain that you've got an issue with how the methodology was put together. It might be a study that demonstrates 50% of people got better with da-di-da, but there was only 2 people in a study.
Yeah.
So you need to be able to critically analyse research, and most unis now teach how to do that, but it is still very confusing, and that's where did you go through New Zealand College?
Yeah.
Yes.
I mean, I had Dr. Heidi as my neuro
and she's
teacher anyway.
Yeah.
But I guess it's one of those things that I found a bit harder to learn because I went back at 33, and it's not the way that I've had to think for so long. And I've read all those articles you sent through on the drive anyway, and I was a little bit like, well, I kind of get it. I understand the abstract, but I don't get statistics that are great. I never got statistics very well as pre-chiro anyway, so I'm just kind of figuring out. I get the understanding behind it. I understand that what they're trying to come away with, I guess, but the actual understanding of it is one of the things that I generally as a whole struggle with, because it's kind of a little bit foreign to the way I think anyway. So I just wondering if there was a more, I guess, layman way of figuring out how to look at these things or
Yeah, well, I hope that what I've just gone through was layman enough for you.
Yeah, definitely.
In quite, little snippets of information, what an entire article, because believe it or not, the first article I wrote was 8 pages long and it took 2 years to write.
Yeah.
To get the information, to review 60 years worth of data, to compress it, to analyse it, to statistically analyse it, to get graphs, to make it all accurate, to submit it 16 friggin' times, and then it gets accepted to be published. So it takes a lot of work to whatever words they put on that page are really thought of very specifically. And so we can do more of this kind of stuff. But again, Heidi's stuff where she changes it into animations may be something worth looking into.
Yeah. No, I mean, I'm always interested in the data because I do enjoy the critical look at it. It's just statistics are just. I've always looked at statistics as an opportunity to use numbers to back your own argument, one way or the other. So I always find it interesting trying to figure out where they're trying to come from with particular sets of data, and is it clinical? Is it statistically significant or not? I guess is the other way of looking at it. I mean, if you were to do more kind of journal article reviews and things like that, that would be awesome, actually.
Yeah, well, it's all this stuff that I've just given you, and all of those articles have other articles that attach to them.
Mm-hmm.
But again, it's just practicing. It's just doing a little bit at a time and doing more. We've actually got a research for the novice investigator course that I wrote with a lady that was working with me a couple of years ago.
Mm-hmm.
It's on our Todd Education portal. So if you need CPD points, you get all your CPD points for the year, and it just gently takes you through how to do research, how to read research, how to write research, like to do a case study.
Yeah.
With little videos and things like that. So, and there's a percentage of all of the money goes to the Chiro Association and ASRF. So there you go. Okay, any other questions? All good. Well, we timed it very well, I think. We're right at the death knock as far as the time goes. Thank you very much for coming on. Appreciate it. And I know that in a couple of weeks' time, I think Craig is doing another one. Wayne and I will be away for a little while. We're going overseas, and I'm actually doing a presentation on my research at the Edinburgh Lectures, and Wayne's doing a talk on SD protocol. So we'll be away overseas, but of course you've got the lovely Justin, the team at the support office looking after you, and also clinically Craig will be about as well, and some of our senior chiros I'm sure can be someone to give a call to if you've got any burning clinical questions. Okay, take care.
Starting onto that
Thank
one.
you, guys.
Thank you.
Bye.
Thanks, Ed.
Thanks. Bye.
Yeah. I don't think most people are aware of Dan Murphy's stuff.